Good afternoon. I would like to welcome everybody to this Fourth Monday Colloquium. Dr. Stovall is going to give us a talk. The title will be The War on Cancer. All's Not Quiet on the Wesleyan Front. Before we start, I would like to introduce Dr. Stovall. Dr. Stovall graduated from Campbell University in 2009 with a BS in biology, graduated from Wake Forest University in 2014 with PhD in cancer biology, and joined our faculty in 2014 as an assistant professor of biology. He teaches human anatomy and physiology, developmental physiology, histology, pathophysiology, and cancer biology. He also serves as an advisor for the Science Club and a program coordinator for the biomedical science major. So without any further ado, let's welcome Dr. Stovall.
Thank you. All right, so good afternoon. Thank you for coming today. Appreciate that. So I picked this title because there's this interesting revival of this war on cancer that's been taking place just this year with the Obama and Biden administration. And the goal is really to eradicate cancer, to build on this initial foundation that was started in the early 70s by the Nixon administration. And I like this, the book, All's Quiet on the Western Front. One of the most depressing books I ever read in my life. I hated my 10th grade English teacher for making me read it, but it was actually a really good read. I should note that a lot of cancer scientists don't like this analogy of using the idea that we're going to war with a disease. And the reason for that, aside from the personification of a medical condition, is that in war, there's always collateral damage, right? That's the one thing that happens in every war. And what you want to do with a patient is not to collateral damage your way to the eradication of the disease by harming all these other useful normal cells, but rather to find specific molecular targets that your cancer is dependent on, the Achilles heel of every single cancer cell within a tumor. And that Achilles heel may be different for one cell type versus another. So as we go through this concept of this war on cancer and the role we're playing in it, I don't want you to consider that we're trying to nuke it. We're trying to go in like special ops and find particular aberrations for each cancer cell that we can exploit therapeutically. Any excuse to use Latin, I'll take it. Dice quasi victurus, vivi quasi cras moriturus. I like this because it says learn as if you will live forever and live as if you will die tomorrow. So we'll talk about this. We'll begin by looking at the prostate, which is incursion site 0. This is where the tumor actually develops. For our purposes, we're going to study prostate cancer for a list of reasons I'll give you in a few moments. And from there, we're going to begin studying not only the organ in which this tumor develops, but a little bit about the pathology of the disease. It is an unprincipled and unscrupulous enemy. And then we'll look at SOX17 and the role that we think this gene has to play in this ongoing battle. And finally, where we go from here, sort of the future plan of research for our lab. So we'll begin by looking at the prostate. So I'll warn you, this is what my anatomy students have to endure, is these somewhat graphic images. This is the male reproductive tract. The prostate is indicated by the blue arrow. It sits just in front of the rectum, just beneath the male bladder. And the normal function of this gland is really to release fluids that contribute to semen volume in the male reproductive tract. And about a third of your semen volume comes from prostatic secretions. This gland tends to grow over the course of a male's life. It continues to grow. In most men over the age of 50, really, and especially over the age of 60, you'll see the prostate has begun to enlarge. That's totally typical of the prostate. An enlarged prostate is not a cancerous prostate by necessity. But for sure, growth in any tissue does predispose it to become cancerous. So some of the things that this gland releases into the male reproductive tract are enzymes such as prostate-specific antigen and nutrient sources. Those are basically to help the sperm survive the very hostile environment of the female reproductive tract. So sperm will be ejaculated up here. They'll come through this long duct into what's known as the urethra. This is the passage that sperm and semen and urine will all take out of the male body. The prostate sends its secretions here. So this is one of the first semen joining sites that sperm encounter as they enter into this tract. So what happens when this organ becomes cancerous? Well, prostate cancer is a pretty important disease to study because of its prevalence. Over 180,000 new cases are predicted this year, and again, over 26,000 deaths projected by the American Cancer Society just this year. Nearly 3 million men living worldwide with prostate cancer at present. This disease is typically pretty slow growing, despite the fact that this is the most common cancer in U.S. men, aside from skin cancer. It is only the second leading cause of cancer-related deaths in U.S. men. If you look at prostate cancer patients, if they're diagnosed with prostate cancer, while that tumor is still localized to the prostate, or even if it's still localized to nearby lymph nodes, they have nearly 100% of those men will be alive 5 years from the time of diagnosis, which is a great survival rate. Typically, an older gentleman goes into the oncologist's office. The oncologist with prostate cancer, the oncologist is going to think, this guy's going to be a cardiologist's problem, or he's going to be a nephrologist's problem long before he's my problem. So prostate cancer, the typical treatment plan for these presentation with early disease is a watch and wait approach. However, if a male presents with the disease, after that disease has already spread to other sites of the body, that 5-year survival rate plummets down to less than 1 in 3. So we have a 28% chance of survival 5 years out from diagnosis when we have distant disease, and so the disease has spread. So one of the major issues to understand about prostate cancer in this war is how this disease spreads, how it moves from one site to the next. Because over 90% of cancer-related deaths are not due to the primary tumor. A man can live without his prostate. A woman can live without a breast. But they can't live without the bones and the lungs and the brain that those tumors spread to. Okay? So understanding that metastatic process, that process by which cancer spreads, is going to be very important. So fundamentally, the enemy here is cancer, and exactly what is it? If you'll think back to your Bio 101 days, this is a figure that comes from our Bio 101 textbook. So the cells in your body are continuously cycling through various states of being. They're either growing and replicating their DNA and growing, and they're doing their cell stuff. They're performing the activities in the body that they were meant to do, or they're actively dividing and producing more cells like themselves through mitotic cell divisions. So cancer is a set of diseases. Never think of this as one single disease. It's a whole mess of diseases, and they're characterized. One commonality in all of them is that cell division has become deregulated. So now cells are not concerned with carrying out their normal functions. They're not stuck here in G1 phase. They're not standing still, doing their cellular activities, carrying out the normal functions of the organ they're making up. Their sole purpose in a cancer cell is to make more copies of themselves. So they get caught in this loop, and they're blowing past checkpoints that are supposed to be stopping cells from just cycling and cycling and cycling through this cycle. This is what two cancer cells two cells look like that have just undergone cell division. This comes from a lab up in Oxford. And so this is what you would call fluorescent microscopy. These cells have been stained. The DNA in the nucleus is stained blue, DAPI stain, and then different cytoskeletal proteins that make up the structural backbone of the cell are stained in red and green. And so this is what it looks like after that one cell has just split into two cells. So, of course, cells have normal functions inside of an organ, right? Your prostate is an organ. It's made up of lots of different types of tissues. It's made up of tissues that secrete things. It's made up of tissues that are muscular, that sort of squeeze. It's made up of tissues that are connective, that sort of support and bind that organ together. Each tissue, then, is made up of different types of cells. And so to understand what any normal cell is supposed to be doing, you have to understand a little bit about the goings-on inside of that cell. So I want to focus on this concept of gene expression. So any cell has DNA in its nucleus. Any eukaryotic cell has DNA in its nucleus, and that DNA contains the genetic information for all of the activities the cell should perform. But it's not readable. It's an illegible code the cell cannot interpret. So the cell rewrites its DNA instructions as RNA. They're both nucleic acids. They're in the same language. It's just a legible version of that genetic code. The messenger RNA then gets translated into the language of proteins. And those proteins inside of the cell and outside of the cell and on the surface of the cell are what actually carry out the cell's activities. And they allow it to do all the things that it's supposed to do, including dividing. And cell division has a very important role in the normal scenario. In a normal healthy tissue, if something gets damaged, cells have to be replaced. You need the cells to be able to divide, right? So there are genes, there are systems in place already that predispose cells to that cell division behavior, and those genes can be turned on. You can express that genetic material as proteins. They can conduct their activities in the cell and promote the activity the cell needs to perform at the time. But it's all very regulatable. In a cancer cell, this process of gene expression has become derailed. So broadly, we can classify these genes into two groups: tumor suppressor genes and oncogenes. So I'm going to talk to you today mostly about tumor suppressors, but I want to compare and contrast them for you briefly. So a tumor is naturally going to want to downregulate or inhibit the expression of these tumor suppressor genes, which, when turned on, antagonize the growth of the tumor. As their name suggests, they suppress tumor growth. These are genes that keep your cells in G1. They keep that cell from dividing. They make sure that it's doing its normal job, and they keep the cell happy, healthy. So tumors downregulate these genes. They don't want those genes on. So if we can figure out pathways by which that tumor antagonizes this gene, and we can eliminate those pathways, then the gene theoretically will be restored, and it will inhibit the growth of the tumor, right? So with tumors and tumor suppressor genes, there's this sort of reciprocal inhibition going on. The tumor doesn't want the gene to be turned on, because if it is, the protein that gene encodes is going to stop the cell from dividing. But the tumor suppressor gene, so there's this reciprocal inhibition. With oncogenes, however, the opposite is true. There's a reciprocal activation. The oncogene promotes the growth of a tumor, which in turn promotes the expression of the oncogene. So in this scenario, if we can identify how the oncogene is promoting the growth of the tumor, we can target those pathways, eliminate them, tumor goes away. Right? So again, we're not looking for sledgehammer chemotherapy techniques here. We're trying to find specific molecular features, characterize these pathways by which tumors are antagonizing gene expression or promoting gene expression, characterize pathways by which genes and proteins are either antagonizing or promoting the growth of the tumor, and target them. So that's our strategy: characterize molecular aberrations and then exploit them therapeutically. This is a hallmark, and I apologize for the poor color quality here. This is a figure from a seminal paper that came out in Cell. It's one of the leading scientific journals in the world. And this was from a famous cancer scientist known as who's named Robert Weinberg. And he presented what he considered to be the 6 hallmarks of cancer. And any genes that are involved in cancer are going to be affecting at some level or another one of these 6 fundamental processes. It's worth noting that in 2011, he came out with a new list of hallmarks. You might imagine that after a decade's worth of research in the 21st century, the list grew. So we're going to consider his original 6 hallmarks of cancers and focus on them, because to look at the new list is a little daunting. So if you look here, one of the things that tumors have the ability to do is evade something known as apoptosis. Apoptosis is programmed cell death. So normal cells, when something goes wrong inside that cell, they try to fix it. If they can't fix it, they commit suicide. So they kill themselves. This is programmed cell death. It's normal. Death is a part of life, all those other cliches. Apoptosis has to happen in order for normal physiology to be maintained. Cancer cells, on the other hand, they accumulate massive numbers of aberrations, and yet they don't die. They have pathways turned on inside them that help them to elude those death signals. Similarly, every cell in your body is in constant communication with other cells in your body. And those cells are continuously telling one another, okay, you need to make more copies of you, need to divide, or no, there's enough of you, you need to stop dividing right now. So there are what we call pro-growth and anti-growth signals. And cells are usually reliant on their neighboring cells for those signals. Cancer cells have basically become deaf to the you don't need to grow signals. And they found a way to produce signals that say, go ahead and grow themselves without having to rely on those signals coming from their neighbors. So cancer cells are quite adept at survival. There's a couple of other hallmarks here I'm not going to really get into today. I'm happy to talk about them. Things called limitless replicative potential, sustained angiogenesis. Our focus is on tissue invasion metastasis, because it is that hallmark that makes cancer so deadly. Cancers, again, 90% of cancer deaths are caused by the metastasis, not the primary tumor. So that is the process that we really want to understand. So I'll introduce you to this group of genes that are called SOX genes. It stands for Sorry-related HMG box genes. Sorry is the founding member of this group of genes. It stands for sex-determining region Y. It's a gene located on the Y chromosome. This is the gene that gives you your maleness. So if an embryo is going to become a male, it has a Y chromosome and an X chromosome. If the embryo is going to become a female, it has 2 X chromosomes. What makes the embryo a male, what kicks off male pattern development, is the Sorry gene located on the Y chromosome. I'm convinced a woman named it Sorry, that you're going to be a man. So this is the founding member of the SOX family. They encode proteins that play diverse roles in development. So these genes are involved in telling cells what to become, what not to become. They're involved in telling cells you need to move over here or you need to go over there. So they're basically helping the embryo to pattern itself and to understand on a cellular level what each individual cell should be in the long run. So they're involved in all of that developmental process. What is a pretty common theme is that cellular programs that regulate development are frequently messed up in cancers. And it's really because a cancer cell has this strange dichotomy. Cancer cells, any cell, is either differentiated and it's doing its job. For example, an immune cell is actually fighting an infection, or a red blood cell is actually carrying oxygen through your tissues, or a cell in the gut is actually absorbing nutrients from your GI. Those cells are doing a job. Alternatively, cells could be dividing. They could be making more copies of themselves. Those cells that are replicative, that are continuously duplicating themselves, they tend to be more stem-like. They're less differentiated, less specialized. And so cancer favors that, because cancer cells don't want to be bothered with doing what they're supposed to do. They want to focus on dividing and dividing and dividing and dividing. And by the way, when biologists use the word cell division. It's the same as cellular multiplication. So division and multiplication are the same in biology for all intents and purposes. When this cell becomes 2 cells, it has multiplied into 2, but it did so by dividing itself. So it's sort of a conundrum. So the SOX gene that we're most interested in here is SOX17. My PhD studies were focused on SOX7, which is a well-known tumor suppressor in a number of different types of tumors. This is the subgroup F. So the whole SOX family is divided up into these subfamilies, A, C, B, D, and so on. My favorites are in the subgroup F. SOX7 is closely related to SOX17, and that's the gene that we study today. Most of these, or all of these SOX genes, what characterizes them, what unites them in this family, is the presence of something called an HMG box. That HMG box is how these proteins bind to DNA. So there are tons of different types of proteins in your cells, right? This particular class of protein are what we call transcription factors, which means that they bind to target genes in the DNA and regulate how well those genes are expressed. Are they going to be transcriptionally active? Or are they going to be transcriptionally repressed? That's what transcription factors do. They target genes in the DNA and they either up or down regulate them. SOX17 and the other SOX members do that by interacting with DNA via that HMG box. And that is what gives them their homology within this family. And as you might imagine, there is more structural similarity between those members of the same subgroups versus different outlying groups. So SOX7 and 17 would be more closely related, more structurally similar than, say, SOX7 would be to SOX15. Okay, so I want to tell you what I know about SOX17, what the scientific community knows about SOX17. We know that it's a tumor suppressor in many cancers. So this is data that was published last year in the journal Oncotarget. And this group showed that SOX17 depletion increases colorectal tumor volume in mice. So this is their data when they implant colon cancer cells into mice, they get tumors that look like that. Now, when they put those same cells into mice with only one change, and that is to have removed SOX17 from the cells, they see the tumors form like this. See a difference? Those without SOX17, much bigger than those with SOX17. So both their weight and their volume increased when SOX17 was lost, suggesting that SOX17 does something to inhibit the growth of the tumor. Similar observation was made in endometrial cancers. If you take a look at this graph, you compare the volume of tumors that were just endometrial cells put in with an empty vector versus those when you knocked SOX17 down, the tumors were bigger without it. And if you forcibly overexpressed this gene in those cells, the tumor size was significantly reduced, suggesting that both overexpression and depletion of this tumor suppressor can alter tumor growth. Similarly, in oligodendrogliomas, which are a type of brain cancer, you'll see a positive correlation between SOX17 expression and cumulative survival. So those patients that have higher levels of SOX17 expression tend to survive on the order of closer to 150, 170 months, as compared to those who have low levels of SOX17, most of them not surviving past 30, 40 months. So this correlates with a greater survival advantage in brain cancers. Now, I told you that tumor suppressors inhibit the growth of tumors. And for that reason, tumors don't want them turned on. They want to silence those genes. How do they do that? Well, SOX17, we know, is silenced at least in one mechanism in breast cancer by a chemical addition to the DNA. We call this methylation of the DNA. So DNA methylation, you simply add methyl groups to DNA bases, and that will silence that gene from being transcribed. So it turns it off. Every black dot represents one base that is methylated in the SOX17, what's called promoter, the region just upstream of the gene, what regulates it on or off. If the promoter looks like this, where most of the DNA is not methylated, the gene is likely to be transcriptionally active. If the DNA looks like this, mostly shaded in, lots of methylation, the gene is likely to be inactive, transcriptionally repressed. Most breast cancer cell lines, here showing 5 out of 7, transcriptionally repress SOX17. They modify it chemically on a genetic level to try to repress it from being expressed in those cells. So all of this led us to the question, what's it doing in prostate cancer? One of the curious things about transcription factors is that their context can vary from one cell type to another. So just because a transcription factor has one set of functions in this tissue, it could have a completely different set of functions in another tissue. And part of the reason for that is that no transcription factor, or at least no SOX transcription factor, interacts with DNA of its own accord. Its DNA interactions are mediated by other proteins that are around it. Different tissues express different proteins. And so depending on who its interaction partners are, you may have a different response cellularly to one gene or another, in one tissue or another. So we came up with this hypothesis that because all of these we're looking at, with the exception of the oligodendrogliomas, all of these are epithelial tumors, we hypothesized that SOX17 should be acting pretty similarly in prostate cancer. So we think that SOX17 acts as a tumor suppressor and is downregulated by multiple mechanisms in prostate cancer cells. So this really led us into two arms that have been pursued by some great undergrads here at Wesleyan. So one of these questions that I'll talk about first is, does SOX17 have tumor suppressive effects in prostate cancer cells? And this work was begun by Tiffany Ramos last year and is being continued by Joanna and Sanabu this fall. And the other question is, how do prostate cancer cells downregulate SOX17 expression? And that is being addressed by Karishma Patel's work. So I'll start with showing you some of the work that Tiffany and Joanna and Sanabu have been doing for us. So does SOX17 have these tumor suppressive effects? Can we see a phenotype in these cells when we manipulate the expression of this gene? Well, that all hinges, first of all, on our ability to manipulate the expression of the gene. So we're most interested in the particular phenotype of invasion. We want to know if it affects so there's a number of different ways that SOX17 can have a tumor suppressive effect. Does it stop the cells from cycling? Does it stop the cells from invading? Does it antagonize a cell death pathway? What exactly it does, what phenotype it has inside the cell, is not well known for the prostate. We're most interested in invasion. So this is the metastatic cascade. You begin with a cell in your primary tumor. It's highlighted for you in yellow there. And that cell has got to locally invade through the surrounding tumor tissue and the surrounding matrix. It then has to intravasate or enter into the bloodstream, where it's going to travel around the body until finally it reaches a site where it extravasates from. It adheres there, it colonizes it, and it forms this metastasis. That's the process we want to stop. You can stop all that other stuff if you prevent that initial localized invasion. The problem with this is that a tumor is detectable at maybe 1 million to 10 million cells. They're starting this process of sneaking into the bloodstream at as few as 10 to 100,000 cells in a mass. So they're already starting to spread before we can even detect the tumor in the first place. So that's a big problem. So what did we do? We knocked down SOX17 in these cells. We used a neat little system called RNA interference in order to do this. This double red line here represents the cell membrane, and this is the inside of the cell. We grow these cells in dishes in the laboratory. They grow at the bottom of a plate. We put media on them. That's their nutrient source. And what Tiffany and Joanna and Sanabu have done is to basically transfect into the cell these small siRNA, small interfering RNA molecules. And what they do is they go through a series of processing machinery inside the cell. All we have to do is put them in the cell. The cell will take care of the rest. They modify these siRNAs, and the siRNAs will target a messenger RNA inside of the cell that they're complementary to. When that siRNA binds here, it prevents this transcript from ever being translated into a protein. So we can stop the expression of the gene at a functional level, at a protein level, through this siRNA system, this RNA interference. And so we do this by designing siRNAs that are specifically complementary to our target. So we can make siRNA, we know the sequence of the whole genome. So it's easy for us to read that mRNA transcript that's going to code for SOX17, come up with the complementary sequence, stick that into the cell's going to knock it down through this siRNA. Then, once the cell has lost its SOX17 expression, we plate those cells into what's known as a Boyden chamber or transwell assay. And so the cells are placed in that upper compartment where they have a very poor nutrient source. And they're separated from a lower compartment that is rich in nutrients and chemoattractants by this membrane that cells can invade through. So our goal here is to basically set up two scenarios where cells that have either control siRNAs that don't target any known sequence are put in, and the cell should have its endogenous, its normal level of SOX17. And secondly, our cells that have had SOX17 knocked down. So now you've got these two groups in the transwells, and you're going to compare the rate of invasion through that membrane between cells with and without SOX17. Tiffany did this experiment once, and then Joanna and Sanabu did it again. Actually, I did it again, and then Joanna and Sanabu did it again. Tiffany, Joanna, and Sanabu's experiments worked. Mine didn't. So they have surpassed me in their scientific technique. So this was the data between their experiments. You can see that there's a significantly increased rate of invasion among those cells that have lost SOX17 compared with those cells that have normal levels of SOX17, suggesting that the loss of SOX17 does promote cell invasion in prostate cancer, which was a really cool finding. Now, I want to switch gears here and talk a little bit about the work that Karishma has done on trying to understand sort of the flip side of this. They're trying to understand how SOX17 inhibits the cell, how it's affecting a phenotype in the cell. Karishma was trying to understand how the cell's downregulating the gene. What mechanisms is it using to silence this gene? So that's the question we'll explore now. Before we do that, I need to give you some background on how cells silence genes. And so the major mechanisms that cells will use, epigenetically speaking, and that is to say, speaking in terms of what they will do to the DNA expression, but not its sequence. So genetics is this really cool field where you study the effects of mutations, which are changes to the DNA sequence. Epigenetics doesn't study how changes to the DNA sequence affect a gene. It studies how changes to the DNA molecule itself or changes to the surrounding environment of the DNA will affect that DNA's expression. So here you have DNA methylation, which I mentioned earlier. This is a common method for silencing gene expression. You'll take these, you'll all remember back to basic biology that DNA has these 4 bases in it, right? What are those 4 bases? Good. A, C, T, and G. We'll keep it simple. A, C, T, and G. 4 letters make up the DNA code. This is what C looks like, right? Now, for DNA, if you simply methylate it, add this one little chemical group to that molecule, and you do that in repetition in the promoter region of the gene, it leads to a tighter conformation of DNA around the proteins that package it. Those proteins are called histones. You actually need those proteins because if you were to take all the DNA in a human cell and stretch it out end to end, it would be as tall as I am. And yet you've got to pack it into a space that's about 5 millionths of a millimeter. You can only do that by winding it and winding it and folding it and folding it. And so this produces this sort of nuclear, this entire nuclear environment that you have to study. So there's enzymes that carry this out. DNA methyltransferases, DNMTs is what they're called. They add those methyl groups onto these C residues in DNA. And when they do, they promote the DNA as being a tighter conformation, which makes it harder for proteins that are going to get in there and read the DNA, express it. They're going to have a more challenging time. So it helps us silence the gene. You can also accomplish the same effect not by methylating the DNA, but by deacetylating histone proteins. So those proteins DNA wraps around, they extend these tails, and often you'll find those tails are acetylated, which is just a different kind of chemical group that you've added onto the histone. When they are acetylated, the DNA is in this sort of open conformation. Gene expression can occur. When you remove the acetyl groups, the DNA clumps up, becomes what we call heterochromatin, becomes tighter, harder to read, and so you get transcriptional repression, similar to what you saw with DNA methylation. So as you can imagine, this was also carried out by enzymes. They're called histone deacetylases because they remove acetyl groups from histone proteins. So there are drugs on the market that are available to inhibit these enzymes. So that was the approach that we took. We pharmacologically inhibited DNMTs and HDACs. The DNMT inhibitor was 5-AZA, 2'-deoxycytidine. We're going to call it 5-AZA. And then SAHA here is HDAC inhibitor. So they're very generic. You know, they basically inhibit lots of different DNMTs, lots of different HDACs, not just one specific targeted protein. So what happened when Karishma put these things into the cells? Well, we measured what would happen when you put these molecules into cells versus when you put a vehicle in, which is just what they're dissolved in. So these molecules are dissolved in DMSO, which is an organic solvent, and so we treat the cells either with 5-AZA or DMSO. And so that's our control and our treatment group. Then we have to measure, does it have a functional effect on the protein level? Does it change how much protein is expressed in the cell? So we do that through what's called a Western blot, which is essentially where you're going to run proteins out on a gel and separate them based on their size. You transfer those proteins onto a membrane, which you can then probe with antibodies that recognize your specific protein of interest. And so they can measure how much protein is in the cells that went into this lane, how much protein's in the cells that went into this lane, right? So this would be your DMSO, this would be your 5-AZA low dose, this would be your 5-AZA high dose, right? And you'd compare the expression of the protein in one lane to the next. You do that on a film, so after you incubate this membrane with the antibody, you've probed it, you're then going to basically record a luminescence on a film, chemiluminescence on that film. You're going to get a pattern of bands. And this is what it looks like. So SOX17 is expressed in control cells at this level. And when you treat those cells with a high dose of 5-AZA, you increase the intensity of that band, indicating that the protein expression has increased as a result of inhibiting those DNA methyltransferases, suggesting that DNA methylation, which those enzymes carry out, does in fact contribute to SOX17 silencing. Similarly, you see this with SAHA. When you inhibit HDACs, the expression of SOX17 also increases, again suggesting that histone deacetylation plays a role in the silencing of this gene. Karishma did this many more times this fall. She worked very hard in the lab this fall to get more data. We ran into technical difficulties with the antibody. The company that we were using to purchase the antibody from for this experiment, which she did last year, has stopped making antibodies. And so I switched to a new antibody. Terrible idea. Now we have lots of technical issues, so I wasn't able to get that data from this fall. Her most recent data isn't included here, but this is what she did last year. It has been repeated. As soon as we work out the kinks with the antibody, I can come back and tell you more about it. But we are happy to say that DNA methylation and histone deacetylation seem to contribute to SOX17 silencing in prostate cancer. So, two broad conclusions. What Tiffany and Joanna and Sanabu have shown is that SOX17 loss increases the invasiveness of prostate cancer cells. And what Karishma has shown is that these cell lines can inhibit SOX17 expression via modifying the chromatin, either changing the DNA or changes to the histone proteins with which it associates. So where do we go from here? Well, there's a lot of future directions for this arm of the project. We're really happy to see that the invasion data looks very promising. But of course, so you showed a phenotype, so what? You need to understand the mechanism. So what is the mechanism of that anti-invasive function? How is removing SOX17 increasing the invasiveness of those cells, right? Characterizing that molecular pathway is important, and so that's a very high priority for the future. Are there other cancer phenotypes? It's not just invasion that's important, although that is the most clinically relevant, but maybe it affects how well that tumor recruits blood vessels into the area. Maybe it affects how quickly the tumor grows, how big it gets. Maybe it affects apoptotic pathways and prevents them from killing cells. So what are those other phenotypes that this tumor suppressor may be affecting? And then finally, we've only done these experiments by knocking SOX17 down in these cells. They're cancer cells, right? So theoretically, they've already got a fairly low level of this tumor suppressor gene. So you're seeing the difference in this level, maybe in this, right? What we would like to do is perform a knockdown in cells that are normal prostate cells that theoretically have a higher level of SOX17 normally. So if you knock them down, you should see a bigger difference, and they should become more invasive when you lose that gene. In the cancer cells, we'd really like to overexpress SOX17. We were going to do that. It requires cloning. Cloning takes time. So we're going to do that. It's on the docket, but we haven't gotten to it yet. And finally, for Karishma's project, it's very important for us to discern whether the effects of those pharmacological inhibitors are direct or indirect. Right? So you put this 5-AZA on the cells, you inhibit DNA methyltransferases, SOX17 goes up. Now, is that because that stopped the methyltransferases from binding to the SOX17 gene and methylating it directly? Or did SOX17 increase because the DNMTs no longer silence this other thing over here, which is now free to go over and activate SOX17, which would be an indirect activation? So we have to tease that apart as to which is it. And finally, there's far more to how cancer cells can manipulate a gene's levels than their chromatin modifications. That's just going from DNA to RNA. Then you've got the whole RNA to protein level of regulation that you have to interrogate. And then even once the protein is made, there are ways the cell can turn it over. So you have to sort of tease apart how the cell's doing that. So all of those are future avenues of research for these projects that we're excited about. So I'll just acknowledge some really important people. Steve Cradell is a PI at Wake Forest University School of Medicine. Amanda Davis is a graduate student in his lab, and they're very generous to us, Pranima Dubey as well. They're very generous. They donate all the cell lines. They donate the media. They help us out a lot. To give you an idea of how much help that is, one vial of cells would cost about $800 to $1,000. One bottle of media, and we run through several a semester, would cost several $100s. So they're saving us a lot of money. Very friendly, very great people. We've been successful to get some research grants from the TLC, so we're very appreciative of those. And then, of course, Math and Science Division budget for letting me chew away at you every now and then. The fantastic undergrad researchers have just been a joy to work with. It's been very fun to watch them sort of grow into these young scientists. That is a crystal structure of the SOX17 protein, if you're interested. So have to give a nod to the little guy. And so I'll leave it there. And I'm happy to take any questions. I appreciate you coming. We know generally how the tumor cell invades. What we don't know is the mechanism of how SOX17 antagonizes that.
Okay, well, so then you said you want to try knocking it down in normal cells
Mm-hmm.
to see if they get more invasive, but if they're normal cells, would they be invasive in the first place?
So normal cells would have a very low level of invasiveness. I say normal, I should put normal in quotes. They're non-tumorigenic, meaning that they don't form tumors in mice, but they are still immortalized and they're grown in cell culture. So they're not truly normal cells, but they don't form cancer, so we call them normal. But so they would have a higher baseline of invasion than a true blue normal cell that came straight out of the prostate. Yeah. Anything else? Yeah.
You may have said this and I just didn't hear it, but when you were looking at the groups, sort of the evolutionary relationships between A, B, C, etc., I know there's a reason why they're clustered together as F, but why did you choose the F, and then what would be the next group that you would look at and why?
Yeah, it's a great question. The sort of the raw answer is that F is what I have the most experience with. SOX7 is a close relative of SOX17. Not all of those SOX genes are tumor suppressors. Some of them are oncogenes, which would promote the growth of the tumor. SOX2, SOX9, SOX10, they're well-characterized oncogenes. So some of the SOX proteins are more well-known than others. SOX17 is one that he's not one of the flashy members of the family like SOX2 is. And so we felt we might have less competition from bigger labs studying the SOX17 gene. Yeah.
Fast forwarding into the future, where would you hope this research in general, I mean, would the hope be to be able to increase SOX17 to the point to where these tumors could be minimalized without radiation, or?
Yeah, so it's unlikely that that we would be able to achieve any sort of therapeutic response by manipulating this gene alone. I think sort of my vision for this is characterizing this gene as whether it could be a biomarker for prostate cancer patients. So if a biopsy is done, you can stain the tissues and see, do they have high, medium, low levels of this protein? And depending on that, they may be more or less predisposed to aggressive cancer or something like that. Yeah, so I think it has potential to serve as a pretty good biomarker. I think the molecular pathways we sort of tease out have a good chance at enlightening us as to some other features within that tumor that we could exploit therapeutically. But in terms of specifically how, I don't know yet. Yeah. I will say that therapeutic exploitation of any weakness in a tumor is only temporary, right? Because cancers are just microevolutionary masses. And anytime, it's like antibiotic resistance, anytime you treat a bacterial infection, there are some bacteria that are resistant to the drug. They're going to survive, they repopulate. Now you treat them with that same antibiotic again, useless. With tumors, it's the same thing. One prostate tumor is not equal to another prostate tumor. Every patient has a unique disease. Moreover, every cell within that tumor may have differences. So this population of cells in this person's tumor may be different and may have different weaknesses than this population of cells in that same tumor. So treating it really becomes a nightmare. So does research. Yeah. Yeah.
You said that the SOX17 gene is a tumor suppressor. You have some of those other SOX genes, I think it was a 9 or whatever, is actually an oncogene.
Mm-hmm.
Would it be possible that the SOX17 is involved in suppression of the...
Absolutely. The network of crosstalk between those SOX proteins is another nightmare that I have fortunately stayed away from for the past 8 years. But there has definitely been work done to show that SOX17 and I think it was SOX2 are in this antagonistic interaction. And so for sure, we know that some SOX genes actually directly bind promoters of other SOX genes. They turn them on and off directly. We also know that they compete for binding with interaction partners, so they can sort of act as sponges and soak up the partners that this protein would need to do its job. So they have a number of different ways they interact. Yeah, yeah. Oh, it is. Yeah, but a fun one. Yeah. Yeah.
Does this have applications to other kinds of cancers, or ask questions that other people, or lead to questions other people might be asking?
Sure. So, yeah, I think ultimately what is going to be attempted is to tease out that SOX interaction network, both developmentally and in terms of cancer disease. And so any insight we can give as to what pathways this particular SOX gene is playing in, I think would be helpful in that long-term goal. Okay. Thank you.